Hello, and welcome back to biology. Today we want to talk a little bit about genetics, and that's going to be genes, alleles, and inheritance. What do those terms mean, and how are they related? But first I want to flash through all the slides really fast. No, I'm just kidding. So the first thing, reproduction is one of the criteria for life. And pretty much at every level, you have some form of reproduction or other. And genes, the DNA, is replicated to produce new copies of the DNA, which means you're getting new copies of genes. Cells divide, producing new cells, and that can come in a variety of ways. It can be binary fission for prokaryotes who are asexual reproducers, mitosis for your somatic cells, and then meiosis, producing egg and sperm for germ cells. And then they also do mitosis too, but anyway, organisms reproduce sexually or asexually to produce whole new organisms. Populations can fragment, producing new population, which actually may lead to, wow, I cannot spell, may lead to differing selective, not selective, pressures and or reproductive isolation. I will change that, a little typo there, in your PowerPoint that you get on Haiku. Okay, so species also may split to produce new species, and that is speciation that arises from that reproductive isolation, which we've already talked about. Okay, so when we look at DNA, so we started out talking about the nucleotides, the base pairs, how they pair up, and then we've got our beautiful double helix, and then it turns out that that coils around these cute little histone proteins, and those make these cute little ball kind of things, and then those start coiling up, and then that coil coils up, and then that coil coils up, and you end up getting the very familiar looking chromosome. This is not the way DNA is all of the time. It's only a certain period during the cell cycle, which we're going to talk about soon. Okay, so these are actual chromosomes, and this is all the DNA that's all coiled up. It's very exciting. And when you look at the anatomy, if you will, of a chromosome, the ends are called telomeres, or telomeres, you can say it either way. The center is a centromere, right? And each arm, when they're connected, is called a chromatid. Okay, so this is a chromosome pair, and this is a single chromosome. However, like I said, when they are connected, each single chromosome is called a chromatid. Yeah, so I know that's a little bit confusing. Okay, so how many chromosomes do humans actually have? Well, you've probably heard the numbers both 46 and 43, right? 2 and 1 are totally wrong. But 48, by the way, are all the other great apes. We have 46 chromosomes, and we have 23 pairs, so the answer to this is 46. Okay, so our chromosomes, we have one from each parent, and so they pair up, and so we get these same chromosomes coming from different sources, and by same, I mean homologous. Each one has the same, kinds of genes on it. It's the same structure, same size, all of this good stuff. And so you have this pair. On each chromosome, there are going to be areas that are called loci, or a locus is one. More than one is a loci. And this is some area that is active in some way, whether it is an actual gene or it's regulatory DNA, whatever. And this banding, these loci, are kind of what we're interested in terms of where genes are on chromosomes. All right, now here's your next question. Mother pregnant with her 4th child remarked, "This one just has to be a boy. It's almost certain since my other 3 children are girls." Which statement best describes the accuracy of the mother's comment? She's right because the genes of the father are dominant over those of the mother. The mother's wrong because there's only a 25% chance that the child will be a boy. I mean, she's got 4 kids, right? The mother's wrong because the chance of having a boy is always 50%. The mother's right because the child usually inherits both sex chromosomes from the mother. Well, this one's interesting. Remember that you do get one gene from the mother and one chromosome, from the mom, and one chromosome from the dad. And when we're talking about sex chromosomes, for humans, that's the 23rd pair, and there are 2 chromosomes that you can get. You can get an X or you can get a Y, right? And girls are XX and boys are XY. So the genes of the father being dominant over the mother, that is not applicable here, right? And really not, actually. And then the whole well, I've already had 4 girls, so this one's going to be a boy, 25%, that's crazy talk, yeah? And then you don't inherit both of your sex chromosomes from the mother. If you did, everyone would be girls. Because remember, boys are XY and girls are XX. And so if you got both chromosomes from mom, girls, all girls, all girls, all the time. And that would not work out either, so that is also crazy talk. But because you have two alleles, one coming from each parent, right, you're always going to get an X from mom, but you have a 50% chance of getting an X from dad and a 50% chance of getting Y from dad every single time. So the mother is wrong because the chance of having a boy or a girl is always 50%. Okay, now let's talk a little bit about alleles. So we've got this idea that genes are a stretch of DNA that that determine traits and things like that. Okay, now genes come in different, kind of like flavors, if you will, and those are alleles. So here you have on this loci, you have the gene for flower color, right? So locus, wow, loci is plural. On this locus, right, you have the gene for flower color. Okay, these are homologous chromosomes, so this is a sexually reproducing thing. But actually, it doesn't matter. They've been homologous. It's not important. Anyway, not important to you right now. Anyway, so you've got these alleles. This one is for purple flowers. This one is for white flowers. So if you have this chromosome, this gene says be white. If you have this chromosome, this gene says be purple, right? Well, that's interesting. So how does that work? Well, genes can have all of these different versions of alleles. It's still the same gene. We're going to look at the dominant recessive system right now. I will tell you right up front, it is not always that simple, okay? But we're going to learn it at the very kind of most basic level. And so we're going to look at some alleles are dominant and some alleles are recessive. If they are dominant, then they can mask the recessive. And we'll talk about some situations like that. But it means they express over the recessive, okay? So dominant, since you have two of them, you could get two dominants, or you could get two recessives. In this case, you will have the recessive trait. In this case, you'll have the dominant trait. And then you could be what's called heterozygous. So these are homozygous, or they're also called true breeding sometimes. So heterozygous, you could have one dominant and one recessive. And the real question is, what happens when you have this situation? Okay, so Mendel, right, Gregor Mendel actually does a whole experiment with pea plants about these traits. And when we talk about something either being dominant or recessive, it's either this thing or that thing. That's called a discrete trait or a Mendelian trait. And when you look at these peas that he had, seed shape, pod shape, flower color, seed color, pod color, and stem length, right, height of the plant, were all these discrete traits. You were either smooth or, round or wrinkly. Smooth or not smooth, I can't remember what he called that, purple or white, yellow or green, green or yellow, and then tall or short, yeah? And so that's kind of how that works, right? So when we talk about Mendelian traits, we're going to talk right now about something called complete dominance. The dominant gene will always overshadow or express over the recessive. So let's look at that little cutie kangaroo. Adorable. It's brown, right? So let's talk about pigment. That one is not brown. That is an albino, yeah? So you have these two different phenotypes, that's the physical appearance, of color, right? So we're going to call the one that has pigment, C for color. We're going to call the one that is albino, little c. Here's the thing. Genes, right, are when you represent them with a letter, the dominant is going to be really whatever the gene is called, and then the recessive will be a lowercase of that. So let's say that you have a gene for, height. You might want to call that, if tall is dominant, you would call it that capital H, and you would call short lowercase h. Okay, and we'll see more of this later on. All right, so that means that we definitely know that we have a little c in the cutie little albino, and we definitely have a big C in the cutie little brown guy, right? The albino has to have both C's, because remember, this is a completely dominant situation, so the albino can't have any allele for pigment. We're going to say that the other gene for this cute little kangaroo is going to be another dominant pigment, okay? So we're going to draw now what's called a Punnett square. We're going to take one chromosome, right? One, and these will be in the gametes. So we're going to take one and distribute, because remember, okay, this is going to be a little bit tricky. So this, let's say this is dad, this little brown guy right here. So brown guy right here is dad. There is a 50% chance he's going to give this chromosome to the gamete. There's a 50% chance he's going to give this chromosome to the gamete. So what we're doing right now is seeing what would happen if we were to actually breed these little guys together, these two homozygous recessive, homozygous dominant and homozygous recessive guys. Okay. Now, mom over here also has a 50% chance of giving one of hers to the egg, and then the other one has 50% chance of giving one to the egg. Okay. So babies will have one of each. So one baby might get this from dad, and this from dad, and this one from dad, and this one from dad, and you might get this one from mom, this one and this one, right? This one and this one, this one and this one. It's a lot easier to see when these are actually different letters, and I'll show you an example of that. Now, when we look at these, we see that all of the babies are heterozygous. What does that mean in a completely dominant situation? Well, it means this. All of the babies here are going to look like dad. So let's take one of these babies, and let's do this again. This is a Punnett square, by the way. So we take one of those babies, and remember that baby was heterozygous. So I think this will illustrate the whole like distributing the alleles to the gametes. Okay, so here's one, here's the other one. Okay, so these are all possibilities. Yeah, 100% of the babies of the homozygous mom and dad are going to be heterozygous. So, but in the heterozygote, yeah, let's drop mom's alleles over here too. And then, or genes, right, because they're each on a chromosome. So dad is going to give this baby, so each square is a baby, remember, this baby, that chromosome, and then he's going to give this baby this one. He's going to give this baby this one, and this to that baby. Then mom is going to give everybody a recessive, because that's all she's got. No, look at this, right? Look at this. You have, in this case, these guys are all also heterozygous, because anybody who breeds with mom is going to have a recessive. They are all going to look like dad. These guys, on the other hand, got the recessive gene from dad and a recessive gene from mom, so they will all look like mom. Okay? So see how that works? So there's a 50% chance of being heterozygous. There's a 50% chance of being homozygous recessive. Okay? Now, let's do scenario 3. Let's take 2 of those heterozygotes and let's breed those together. Okay? So, we're going to do the same thing, distribute the chromosomes, right? Distribute the alleles. Alleles is what they're called. Distribute the alleles, and then we're going to say, okay, we're going to drop down. Now, you'll notice, right, when I start filling these out, okay, I put this one first, right, but dad is a lowercase c, and I put dad first here, but the dominant allele always is listed first. Okay, so you'll never see little c, big C. It's always going to be big C, little c, regardless of who's giving the allele. So dad gave a little one, mom gave a big one, you still write it this way. Now when you look at this, ooh, homozygous dominant, brown. Homozygous recessive, crazy talk, heterozygous, okay, brown. And homozygous recessive, yay, white. So when you breed 2 heterozygotes, you actually have a 3-to-1 ratio of dominant phenotype to recessive phenotype. Okay, and here's just an example with Mendel's flower color, right? So in the gametes, you distribute the alleles, right? So here you've got little p and big P, right? Purple, big P. White will be little p, not W. And you do the same thing, and then we breed these, so these are the true breeders. You get all heterozygotes. We breed the heterozygotes. You get 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. And here's just another example with horses. Horses is going to be the theme of the day for us, okay? So here you get either black or chestnut. This gene controls this color. So dominant is going to be the black, right? Recessive is going to be the chestnut, okay? And so if you have a chestnut dad here, you're going to have two recessives. If you have a homozygous mom, or sorry, I don't know why I keep doing that. I'm so sorry. Heterozygous dad, you're going to have one black and one chestnut, okay? And you'll get the same idea. So these are just other examples of what we were doing with the kangaroos. Here's your question. In the Punnett square below, capital T represents the allele for tall pea plants, and T represents short plants. G represents gray seeds, and little g represents white seeds. What genotype should go in the blank box? Now this is trickier because you're dealing with two traits at a time. And so what I want to do here is just show you what's called a dihybrid cross. This is a Punnett square for the two genes together. And I'll show you one for three genes, and then you can see why things start to get crazy when you have more than that. So anyway, so if you have T, right, and little t, so these are both here, you've got a big T here, a big T here, a little t here, a little t here, and 4 little g's. Okay, and I'll show you how these got here in just a second. But basically, right, if you look here, you've got little t and little t, yep, big G, little g. So the answer is little t, big G, little g. It's like a puzzle, okay? All right, and here's the example. So let's say that you have these two heterozygous horses, right? Big B, little b, big C, little c. Big B, little b, big C, little c. Okay, and we'll talk about what these are later on. All right, so this is dad, this is mom. Dad is usually listed across the top, and mom is usually listed down the side, just by the way. So he has a 50% chance of giving the big B to either gamete, right? So we're going to drop one big B, but he's also got a 50% chance of giving the little b. So we have to do 4 sperm this time, okay? So big B, little b, right? Split those. Big B, little b, because mom over here has the same thing. She's got a 50% chance of giving each of those as well. So she may give a big B, little b, big B, or little b, right? But then you also have to pair them with the C's. So there is a 50% chance here and a 50% chance of this one. So you might get B, and they're independent. So you might get big B and big C. You might get big B and little c. You might get little b and big C. You might get little b and little c, okay? And so we're just distributing the alleles into each gamete, okay? And so you have to do 4 instead of 2 because you have 4 different possible combinations. And then you can see how that works here. You actually, in this case, will have 9 that show the dominant phenotype. And here you have something called incomplete dominance, which is meaning that you have one phenotype for the homozygous dominant, one phenotype for heterozygous, and one phenotype for homozygous recessive. And we'll talk about that in just a minute. Okay, so here's your next thing. That, by the way, was called a quantitative trait. This idea of having more than one gene, and most traits are quantitative, but or continuous. It's called continuous variation or quantitative trait. Most traits are controlled by more than one gene. So in this case, I'm just asking which of these is actually not controlled by more than one gene, which of these is a discrete trait or Mendelian. Height in pea plants, skin color in humans, grain color in wheat, beak depth in birds, or blood type. Well, blood type, you're either positive or negative, right? And you're either A, B, O, right? A, B, O, or AB. So that's actually 2 genes, one for the plus or minus, the RH factor, and one for the actual type. Beak depth, you have lots of different beak sizes, right, in the same species, so that is a continuous trait. Grain color is a continuous trait. You probably don't know that, but I'm going to tell you. Skin color, there's not just 2 of those, right? So that's continuous. But height, tall, short. So height in pea plants. Now for humans, it is a continuous trait, by the way, because they're different heights. It's not just like everybody's short or everybody's tall. Okay, so now let's talk a little bit about that incomplete dominance that you just saw with those horses, because you really saw 3 phenotypes, and that was different than what we had seen before. This is homozygous dominant here, right? and a homozygous recessive. The heterozygote gets some from the dominant, right? But not all of the dominant. It's called a dose, yeah? And so here you get pink flowers. So when you have a complete dominant situation, you will have 2 phenotypes. When you have an incomplete dominant situation, you will have 3 phenotypes, and the ratio will be 1 to 2 to 1. Yeah, it's the same exact Punnett square. Yeah, big R, little r. Yeah. Okay, and here's another lovely horse example because I enjoy those. Here's that E, which was the black or the chestnut. Okay, and so in this case, this is a complete dominant situation. The horse is either black or chestnut. But now you can add another gene, and that is called agouti, right? And agouti only affects black hair. So the trait doesn't actually show up in the chestnut guys. But what it does is it is also completely dominant, and it gives you 2 different phenotypes for black, right? So you either get all black or you get black mane and tail, but a chestnut body, okay? Then you can add what's called the cream dilution, yeah? And so the cream dilution, the CR, that is it has a dosage effect, okay? And we'll talk about that a little bit later on, but it's dependent on whether you had a homozygous or heterozygous up here, and that's a little bit more complicated than we're talking about. But you can see that if you have the cream dilution, yeah, you end up having this light color, right? And so that's how you get the buckskins and, you know, the palominos and all of that. So this is actually 3 genes working together to make all of these different colors. Yeah? Okay, and again, quantitative traits, this is polygenic inheritance. And when we're talking about 3 genes, I just want to show you, this is the Punnett square for 3 genes. Here you have the pod shape, right? You have seed color, okay? And then you have seed shape, all right? And so this gets really complicated. You can imagine 4 genes, you can imagine 5 genes. These are huge Punnett squares. You stop using Punnett squares at this point, by the way. Okay, now there's another situation called codominance, and that is actually going to bring us back to the conversation about blood types. Blood types are not only polygenic, but one of those genes is a codominant situation. And here you have 2 alleles that are dominant, and in this case you see white is equally dominant to pink, and so here the lovely like dark red is codominant to white, and so anywhere that they are expressed, they will be expressed simultaneously. Okay, rooster color is a really good example of that. Here you have the white, which is dominant, here you have the blue, which is dominant, and when you start distributing them, you get this mix of white and blue, right? That's codominance. Okay, and you also see it again in horses, because horse genetics, color genetics, really, really interesting. So here you have this red, right? And so the color here, this one happens to be dominant. This is a different gene than we were talking about before. This one happens to be white, and it is also dominant. And what you get is a roan, which is a mix of red and white. If you get really close to this horse, you will see that it is actually a mix of red and white hairs. And you have here a normal mom. Obviously, she has this gene. Dad had this gene, and so you have a cute little roan baby. And then here is a blue roan. This is where instead of having red, you have black. And this black is dominant, again with the white, and so you get a blue roan. By the way, the red roan, also called a strawberry roan, so cute. And then here I just wanted to show you, it's not just 3 or 4 genes, it's lots of genes. So now you can start adding in spotting and, you know, all kinds of things. You can start adding in leg colors, and it just gets crazy. All right. Now, the last thing I want to talk about really quickly is pleiotropy versus polygenic inheritance, okay? Polygenic inheritance means that many genes control one trait. However, there are some genes that control or participate in many traits. So pleiotropy is the opposite of polygenic traits, right? These continuous variation. Some biochemical pathways share genes. So if you have a mutation in one of those genes along the pathway, or you have a change in one of those genes along the pathway, you may find that you get changes that you did not expect. And here's a really good example. This little kitty here is deaf, but only in this ear. Okay, so you can see that she has two different eye colors. Well, the blue eye, okay, if the cat is white, yeah, blue-eyed cats that are white have a 40% chance of being deaf. So the white color in this is the result of what's called dominant white, or it's sometimes called white masking. It's a gene, and so it causes the cat to be white, but it also causes the color of the eye to be blue, right? And I also put this in here because you can see that the eyes actually can be different colors because they are two different, structures. I mean, they're the same kind of structure, but they can be independent of each other in terms of color, okay? So anyway, so this little blue, but it's on the same biochemical pathway that hearing, some of the functions for hearing are on, and so you get a little deaf kitty, right? Okay, so that is it for our conversation about discrete traits versus continuous variation. Our next conversation about genetics is going to be about the whole population and alleles within the population changing, natural selection's effect on alleles, things like that. Okay, so that's it. Thanks for listening, and I will see you next time.